Okay, so Vijayan, you can start. Satish, you can unmute and start.
Yes, yeah, I'm so. Thanks, Abhishek. Yeah, 1 second. Very good evening to you, all of you. And my name is Satish, and I'm one of the organizing members of the Science People. And we are based out of Delhi, you can say, but it's an online pan-international working. We are closely with all the eminent science personalities and science enthusiasts. So Science People started this about 2 years ago, and we had conducted about 52 talks on different topics in the part of science. And this is a second such commemoration of the prominent Indian scientists we are doing. And first we did last year for Tanu Padmanabhan. This year, we are doing for M. Vijayan. Actually, we feel more grateful and proud, and we really want to celebrate his work. One of the legends of the Indian science, especially from the Malayali communities. But really, we can see him beyond that. So this is a 1 hour program, and we have speakers. I welcome all the speakers: Professor Rajan Gurukkal, Professor Murthy, CG Suresh, and Sri Saikrishnan. And we have our moderator, Mr. T. N. Narayanan, and we have some other prominent guests from different institutions. We all welcome everyone, and we request them also, once the talk is over, you can also share 1 or 2 memories or some of his contribution in case you want to for a 1 or 2 minute, and after the speakers sessions. Yeah, and this is a very interesting, and we really hope it will be one of the very important moment for us. So I request Narayanan to take over and invite the speakers. And thank you for that. Once again, I really thank all those participants who joined. Yeah, thank you.
Okay, thank you, Sadishagan, and good evening all. Today we have a very tight schedule with eminent speakers lineup. Let me give a very brief introduction about this program before going to introduce the speakers. So today we are commemorating the contributions of a gem of a person, Professor Mamunumana Vijayan, or simply Professor M. Vijayan, who was with us physically until the last month. He was a person who could associate with several laureates and distinguished fellows and scientists within his domain and out of his domain. And also he was a person initiated the research on macromolecular protein crystallography quite intensely for the first time in India at molecular biophysics unit, a department which is founded by none other than Professor G.N. Ramachandran, another an outstanding figure in the field of structural biology. Ramachandran plot is one of the things still biophysics students study in their classes. So both of them were Keralates by birth. That's another, some of us are proud on that. And but GNR was mostly at Madras, like another Ramachandran and MGR. Both of their workplaces were in Madras. But just to coin the importance of the structural biology or crystallography in biology. Apparently, I just want to mention that Vijayan was a trained physicist, and so he was doing the chemistry aspects of macromolecular molecules or biomolecules those who are interested in medicine and related fields. So just to coin its importance of this field, at least 13 Nobel Prizes were awarded from 1946 to 2009. When the last time Vengi Ramakrishnan was Indian-born scientist, Professor Vengi Ramakrishnan received the Nobel Prize for structural aspects of biomolecules. So Professor Vijayan was not just a scientist, he was beyond that. He was a polymath and multi-potentialite. So we are fortunate to have his close associates, students, and friends who are ready to talk to us today. And let's hear from them more about him and his work. So we have first Professor Rajan Gurukkal. I don't think Rajan Gurukkal needs any kind of introduction to any audience, particularly to this audience, but just for the sake of formality, let me introduce him to you. Because Rajan Gurukkal is a well-known historian, social scientist, writer, and he was a former vice chancellor at Mahatma Gandhi University. He was a visiting professor at the Center for Contemporary Studies at IISc. And from there, I think his intense friendship with Professor Vijayan started. And even his own words of Professor Vijayan, Professor Rajan Gurukkal is one of his closest friends. So with all respect, let me invite Professor Rajan Gurukkal to share a few words and his experience with Professor Vijayan and his interactions. Thank you. Professor Rajan Gurukkal.
Yes. Am I audible?
Yes, sir.
Okay. Respected Professor M.R.N. Murthy, other fellow speakers, Dr. Satishan and others, the organizers of the Science Society. It's immensely gratifying to associate myself with this forum as a friend of Professor Vijayan. As you know, I do not know the actual science about his research, but very generally I know that his contributions have been in the field of macro molecular biology and crystallography. We have Professor Murthy here to educate us in the field. My relationship with Professor Vijayan has been personal, and I used to visit his department ever since my joining the Center for Contemporary Studies in Institute of Science. I remember the very first day of my joining the Center for Contemporary Studies. Professor Vijayan gave a call to the office asking about me, and then we conversed for some time. He said he had heard about my joining the department. Professor Raghavendra Gadarkar had informed him of the date of my joining, and he was so humble and thought of calling me and then conveying his regards. See, compared to Professor Vijayan, I am nowhere. But still, he showed, great admiration and so on. And whenever I used to sit with him, he has been reminiscing his student days in the Kerala Varma College and his active involvement in student politics. And every time I visited, one episode or the other he would share. And at some point of time, I thought that it would be very good if he had written down all these. And one day we discussed about the memoirs, writing the memoirs. And then I requested him to do it in Malayalam first. It so happened that I was asked to write the preface to it. Not preface, an introductory to the Malayalam version of his memoirs. And I am proud of saying that I am one of his admirers who assiduously persuaded him to do it in English. And then finally that also happened, and then it came out as a coffee table production by INSA. I was really happy to receive a complimentary copy of the English version too. I never had a scientist in my conversation sharing the political economy of science, political economy of scientific research. He has been giving me a lot of insights in doing a work on history and theory of knowledge production. I had to deal with production of science from the Renaissance period to 21st century. And I discussed various aspects of the book with him. And finally, when the Oxford University Press published it, Professor Vijayan was kind enough to give a very, very general, very, very generous review of my work in current science. So all that I have to convey at this moment is my indebtedness to Professor Vijayan. And I don't want to take much time because we have other speakers who would share with us the academic research and then various aspects of collaboration. Particularly, Professor Murthy would be able to tell us how he has been associating himself with him academically. And that is going to be really the educative work. Now, I just remember one event in his department. By the time his motor neuron disorder has been troubling him, and still he was able to keep himself academically alert. And then my presence, as I said earlier, encouraged him to switch over to the politics of science. So one day we had a very long discussion on how the dominant economy today, the current dominant economy has been heavily depending on science and technology for capital accumulation. So he inspired me to think seriously about knowledge economy, which heavily depended upon science and technology for the production of marketable knowledge. And then area, the crystallization, crystallography, and how knowledge economy has been using robotic crystallization procedure, and also he discussed micro array technology and so on, and how they're using young scientists for the production of marketable knowledge. He was aware of the large number of huge research establishments built up globally by the corporates for the production of new knowledge and for the production of various other aspects of knowledge like intellectual property, patents, and so on. how these were exchanged at huge rates, and how the capitalist system after the 2008 recession was able to recover in such a way that it would never face recession again because of its dependence on production of knowledge. Production of new knowledge would mean that it would be free of what is called the law of diminishing returns. Economics used to tell us about capital accumulation facing the law of diminishing returns over the years. But once knowledge happened to be its commodity and also capital, then it got released completely from the problem of diminishing returns. So that kind of capitalism would get completely globalized in a hegemonic way. So we used to discuss that kind of politics for quite some time, even when he was not able to lift his face properly because of the motor neuron disorder. Now I often feel really sad when I reminisce such occasions. Now I don't want to elaborate because that will make me upset because of personal reasons. I don't have anything else to say at this moment. Thank you very much.
Thank you, sir. Thank you very much for sharing your valuable memories. And I think I should mention that we are all fortunate to have that Vijayan's memoir. I could go through it, and it was a very lucid piece of work with in all aspects. So I request you to continue in this platform until we finish the program. So some of the participants may want to ask you some doubts or some of the aspects of Professor Vijayan. So let's go to our next speaker, Professor C.G. Suresh. First of all, let me thank Professor C.G. Suresh. He was quite enthusiastic when we approached him. Quite enthusiastically, he supported this program, and he only directed us to make this program today and directed us how to conduct this program today. Professor C.G. Suresh, he was a scientist at the National Chemical Laboratory, Pune. He was one of his first students of Professor Vijayan, to my knowledge. And before he moved into macromolecular crystallography, I think he was mostly working on small molecules, and Professor C.G. Suresh was one of his students who worked on these small molecules aspects. And with all respect, let me invite Professor C.G. Suresh for his words and scientific discussions. Professor Suresh.
Thank you, Narayanan. Can you hear me?
Yes, sir.
Professor Murthy, Professor Rajan Gurukkal, Professor Saikrishnan, and dear friends, very good evening. First, let me thank the organizers of this program, members of science people, Sadishan, Narayanan Tarangat, Narayanan Karur, Francis, and others for giving me an opportunity to present my tributes to the fatherly figure, Professor Vijayan, as his student. My intention is to introduce the life and career of Professor Vijayan, how he bloomed into a great scientist, and more than anything, how he remained a compassionate human being. I shall present a part of his contributions in small molecular crystallography. Professor Murthy will present more about his contributions to science administration and macromolecular crystallography. If I can share my screen.
Yes, you can.
There is a share screen near chat.
Yeah, down.
At the bottom of the toolbar.
Yeah.
with an up arrow, right?
It's already shared. Shared.
No, no, sir. No, sir. So near to this chat box in the down of the panel, you can see a green share screen. You have to click that, and then desktop. You have to share the desktop.
Visible if you bring the cursor down.
Yeah. Yes.
Now it's coming.
Yes. Yes, sir. Yes, yes, sir. Yeah.
Thank you.
Very perfect.
Is it okay?
Yes, sir. Please go ahead.
Yeah. So I have depended on his autobiography for materials used in the slides. He has narrated to us about many of the people and incidents at various times. You can know more about him from the 3 books I have listed here. So this slide shows some of the books listed about him. So he was, Professor Vijayan was born as the eldest son of Subramanian and Uma Devi. He has a sister, Mira, and 2 brothers, Devendran and Surendran. After MSc at Allahabad University, he joined for PhD in the physics department of IAC. The physics department was established by, C.V. Raman, and that time the head of the department was R.S. Krishnan. In fact, R.S. Krishnan was hailing from a village near to Professor Vijayan's place. So that is also one of the reasons he joined the physics department, even though he had other choices also, like joining G.N. Ramendran, etc. So Vijayan chose to do research in crystallography under M.A. Vishwamitra. And crystallographic, that is, the crystallographic studies in the department were established by G.N. Ramendran and his younger colleague Gopinath Karta in late 1940s and early 1950s. When they moved to Chennai in 1952, S. Ramacheshan took over the X-ray work in the department. He continued till 1962 when he moved to IIT Madras. Both Ramendran and Ramacheshan were the students of C.V. Raman. And M.A. Vishwamitra was the student of Ramacheshan. So you can see the lineage, grade lineages of Professor Vijayan. And Professor Vijayan worked on metal complexes of antipyridine and analgesic and some ferromagnetic materials also. Then Kalyani Vijayan joined as his junior and worked in the same laboratory. For the most important center of crystallography and molecular biophysics in India then was the department in Madras University, founded and nurtured by G.N. Ramendran. GNR had great influence over Vijayan's research career. GNR started the annual National seminars on crystallography, which continues even now. In 1967, GNR organized a major international conference. Among the distinguished chief guests were Linus Pauling and Dorothy Hodgkin, both Nobel laureates. Dorothy was awarded the Nobel Prize in 1964 for the structure determination of vitamin B12. Vijayan met Dorothy Hodgkin for the first time at this conference. Dorothy already heard about Vijayan from M.A. Viswamitra and also examined his PhD thesis as an external examiner. She offered to take Vijayan in her laboratory at Oxford, UK. There were two groups working in Dorothy's laboratory. One concerned with the analysis of small molecules and the other on the structure of insulin. Dorothy left the choice to Vijayan which group to join. Vijayan's well-wishers advised him to join small molecule group because work on insulin had not till then produced any definitive results. However, Vijayan decided to join insulin group that turned out to be fortunate. When Vijayan joined the insulin group in 1968, the senior most members of the group were Guy Dodson and Eleanor Dodson, who started working from 1960. Guy was from New Zealand and Eleanor from Australia. Other members were Ted Baker and Tom Blundell. Ted Baker again is from New Zealand, and Tom Blundell took his PhD at Oxford from England. When Kalyani finished the PhD, Dorothy got her also to the Oxford lab. Vijayan and Kalyani got married on 14 July 1969. Dr. Kalyani Vijayan started work on a cephalosporin analog with Dorothy. They left England in 1971 after successfully completing the insulin work. Dr. Vijayan and Dr. Kalyani visited again in 1976-77 in connection with the retirement of Dorothy in September 1977. Their daughter, Devi, was born in May 12, 1977 in Oxford, coinciding with the birthday of Dorothy herself. Owing to our reference to all involved in Devi, we sometimes call her Devayani Kalyani Dorothy Vijayan. When Professor Vijayan came back from England in 1971, initially he joined the physics department and Dr. Kalyani joined National Aeronautics Laboratory, Bangalore. Subsequently, in 1973, at the invitation of G.N. Ramendran, Vijayan joined the newly founded Molecular Biophysics Unit. When I joined Professor Vijayan's lab in 1980 as a PhD student, there were 4 crystallographic groups in IAC. One was in physics department under Professor Viswamitra. Professor Viswamitra is shown here. Then second was in organic chemistry under Professor Venkatesan. Professor Venkatesan is shown here. And third one was in inorganic and physical chemistry headed by Professor H. Manohar. And other than the fourth one was Professor Vijayan in Molecular Biophysics Unit. Professor Vijayan was the junior most. Other than Viswamitra, the 3 professors were combinedly giving crystallographic courses and separately advanced specialized courses. We students invariably attended all the crystallographic courses and used to have interaction with all the 4 professors. Unfortunately, now none of them are alive. MBU faculty consisted of V. Shashishekaran here, then VSR Rao here, then C. Ramakrishnan here, M. Vijayan, then Balaram, and KRK Iswaran here. GNR became an independent professor outside MBU at that time. We all looked at GNR with great reverence. GNR is shown here, sitting with Dorothy Hodgkin when she visited IAC. That time we were learning about his contributions, GNR's contributions to the understanding of peptide conformation, and the celebrated Ramendran plot, which many of my colleagues will be knowing, and they would have studied also. This is an invaluable tool for protein structure validation, the Ramendran plot. By the time I left MBU in 1987, the faculty expanded to include Dr. A. Sarulia. Already Saraswati Visheshwara was also there. Then Samir Brahmachari, not in this photograph. Then Manju Bensal and Dr. Professor M.R.N. Murthy. So these were the faculty members at the time when I left in 1987. When I joined the lab as for PhD, those present in the lab, or those present, the students present in the lab were Sudhagar, Krishnamurthy, CMK Nair, Dinkar Salunke, Francis, and Thanraj. And when I left the lab in 1987, the additional students who joined in course of time were Veerapandian, Kodandapani, Shekhar Mande, Jayanti Ramaswamy, Madhusudan, Jayasri Soman. And Dr. R.V. Tamban was there with an independent project. Nagaprabha was project assistant. And the students who did PhD before me there were P.P. Singh and T.N. Bhatt. And those days, Professor Vijayan used to be in the lab before 8:30 in the morning. He generally expected all of us to be around, if possible, with some new results. Those days, IISc had a DEC 10 computer. Our submitted computer jobs used to run in the night, and we get results in the morning. So we could interpret the results in the morning and present him. This is also the reason for his expectation to see new results first in the morning. We will have opportunity to discuss with him extensively then in case he goes away for teaching or any administrative work. Each day he expects every student to meet him sometime or other. Dinkar used to be the first person to be in the lab, and Francis used to be the last person to leave the lab. There used to be only a couple of hours between these two events. The lab will have full quorum on almost all days, including the weekends. Students very rarely take long holidays. Our experiments involve first growing single crystals of the molecule or molecular complex we are interested. To collect the diffraction, we use a setup as before going to that, these are some of the members when I was in the lab. Myself, Dinkar Salunke, Dhanraj, Veerapandian, Shekhar Mande, R.V. Tamban. So this is the setup we used to use for collecting X-ray diffraction. There is an X-ray source, then the beam is collimated onto a crystal. The crystal is kept here, and the diffraction beam is collected on a photographic film or a detector. There we used to use photographic film. In the lab, there were two setups for recording X-ray diffractions. So one is called Weissenberg camera, shown here, and the other is the precision camera. These two were available during those days in the lab. And the pattern, diffraction pattern we get looks something like this. For Weissenberg in the left side and for precision on the right side. There is a more advanced automated computer controlled data collection setup, which is a diffractometer. So a sketch of that, where the crystal is kept here and the X-ray beam is from here and a detector is here. So this detector goes around collecting the data. Once we index the crystal, then it can be oriented in such a way that the X-ray beam reflects into the detector, and the detector records the intensity directly. That is one advantage with that. That time there was only one X-ray diffraction, the diffractometer. The time we had only on one, and then the diffractometer time was so precious. The crystal could be directly put on the diffractometer to characterize and continue data collection. That is possible. But determining and confirming unit cell can take more time and sometimes go wrong also. So Professor Vijayan, to save diffractometer time, always insisted on us characterizing our single crystals on a Weissenberg camera in the lab before going to the diffractometer. Professor Vijayan goes into the details of crystal characterization before going to the diffractometer. Also, if we have arranged to go out of station and if diffractometer time comes in between, then we have to cancel everything and stay back to utilize the diffractometer time. Most of us used to be present in the lab on Sundays and other holidays. Professor Vijayan also will be in the lab. He used to take us to the canteen for tea on those holidays. During the visit to canteen, Professor Vijayan will discuss with us several issues. We can also hear stories about people and places of his life. That is also the time when we can ask about anything related to science, research, or anything under the sun. Even when he was involved in several administrative responsibilities, Professor Vijayan made it a point to spend time with students whenever required. Nobody needs to wait for long for discussions on any research related issues. He makes sure that all the facilities will be functional always and are utilized optimally. He even sent a technical person like Govindaswamy abroad to familiarize with the running of an advanced micromolecular X-ray diffraction laboratory. That has helped in establishing and running an advanced macromolecular diffraction data collection facility in MBU. Professor Vijayan got interested in cricket during his post-graduation at Allahabad University. He kept that interest throughout. He used to inquire and update score during cricket matches. He used to be thorough with match details and cricket players. He shares this interest with a few other faculties also of MBU. He was very considerate to everybody, irrespective of their status or position. One incident I remember during the Golden Jubilee meeting of Indian Academy of Sciences held at Bangalore in 1985. He was in charge of accommodation and transport. Many of us students were involved in receiving the fellows and taking them to their respective accommodations. That gave us opportunity to closely know some of the great members of Indian scientific fraternity. On parity, even the drivers used to join for lunch and dinner, etc. The banquet dinner was arranged in the prestigious Bangalore Vidhan Saudha. Because of some regulation for entry inside Vidhan Saudha, the drivers were not allowed inside for dinner. All drivers got agitated and complained to Professor Vijayan. Since nothing could be done about it, he also did not go inside. We students also remained with Professor Vijayan. Thus, he was always considerate the feelings of others, irrespective of their status, and used to support them. Similarly, he treats all students equally, irrespective of whether they are his students or those of others. He makes it a point to attend evaluation meetings, even at remote universities, if the future and career of a known person is at stake. It is the commitment he had for people and science. The projects on small molecules in the lab can be widely classified under study of non-steroid analgesics, complexes of amino acids and other biomolecules, peptides, and ionophores. Proteins are condensation polymers of 20 amino acids. Here shown are amino acid, which has got a alpha amino group, a alpha carboxylate group. There is a side chain which varies for all the for the 20 different amino acids, and there is a hydrogen also for the central carbon atom. And when they condense to form the peptide, as shown here, the carboxyl group and the amino group, they come together and then they form a peptide bond. And this is how a peptide is formed, and then further polypeptide and then the protein can form like that. So that's the interest in analysis of structural analysis of amino acids. Proteins are condensation polymers of these 20 amino acids, and structure and function of proteins depend on a large number of non-covalent interactions. Structural analysis of crystalline complexes involving amino acids and peptides was to determine at atomic resolution the geometrical features of non-covalent interactions. Several crystalline complexes between basic and acidic amino acids and amino acids with biomolecules such as vitamins have been studied. When this work was going on in the lab, Cyril Ponnambrama, the renowned expert on chemical evolution and origin of life, he visited visited IAC, and on hearing his lectures, Professor Vijayan was prompted to look into the evolutionary implications of the work on complexes. It is believed that Earth originated some 4.5 billion years ago. There is evidence to believe that life existed on Earth some 3.5 to 3.8 years ago. The origin of life must have been preceded by chemical evolution, consisted of different stages such as abiotic organic synthesis, sequence-specific polymerization, chiral selection, emergence of the first self-replicating systems. Based on Oparin-Haldane hypothesis and Urey-Miller type experiments under stimulated conditions of primitive Earth suggest that simple biomolecules and other organic compounds could have formed in the primitive non-oxygenous atmosphere with energy inputs from UV radiation, lightning, volcanic eruptions, etc. Here, the setup for Urey-Miller experiment is shown. So formation of polymers from monomers in the prebiotic situation involves condensation without enzymes. A condition for condensation under non-enzymatic conditions is the proximity and favorable juxtaposition of the reacting group. As you have seen in the previous slide, the alpha amino group and alpha carboxylate group, they have to come close to each other so that the condensation can go ahead without enzymes. When Professor Vijayan proposed this idea, it received enthusiastic support from biologists such as Pushpa Bhargava and John Barnabas. Here I have to mention that Barnabas was working on biological evolution at that time. By the initiative and encouragement of Professor Vijayan, I joined Barnabas at NCL. I had the opportunity to collaborate with him and his wife, Dr. Shama Barnabas, in the study of human populations and pathogenic mitochondrial genetic mutations using mitochondrial DNA. Analysis of several available crystal structures containing amino acids also showed that they aggregate in a head-to-tail fashion. Some of the, that is like what is shown here, the different types of head-to-tail sequences. Here this amino acid, carboxyl group, amino group, next amino group is here. Like that, it will be, this pattern will go on. And it is called head-to-tail sequences because the carboxyl group and amino group, they one by one, they will be interacting. So these are different head-to-tail sequences in different crystal structures. This what is called a straight, then zigzag, then DL head-to-tail sequences. So in crystal structures, when we analyzed, there are certain patterns of aggregation. One is shown here, is the straight arrangement with you can see the carboxyl group here, amino group here, and the these molecules are translated from one to the other, like that. It will be, this pattern will be going on in two directions. Similarly, these are all generally found in the hydrophobic amino acids. And in the hydrophilic amino acids, this type of patterns, that is one straight and another is the zigzag sequences. These are the patterns which you have found in crystal structures. And also several available, some patterns of amino acid aggregation, that is what is shown here. Similarly, chiral selectivity is shown using crystallization experiments mixing D and L histidine with the D and L aspartic acid molecules. So the molecules of the same chirality, like L aspartic acid and L histidine, or D aspartic acid and D histidine, only used to form complexes, not the L histidine and D aspartic acid. Such type of complexes never used to form. So this also shows about the chiral selectivity. Then in the supramolecular complexes of basic amino acids with the succinic acid, demonstrated the relation among aggregation, stoichiometry, and ionization, which is shown here. The different ionizations of succinic acid, there are two groups which can either be ionized or not ionized, and alternatively one or the other. And there is a hydrogen bond between two molecules, and the other two can be ionized or non-ionized. And the different with the amino acids, in the table it is shown how the stoichiometry changes for these ionization states. So this has been characterized using various crystal complexes, and also complexes of DL arginine with various carboxylic acids showed how, in spite of the differences in the chemical structure of the carboxylic acids used, the aggregation patterns in their complexes exhibit common features. You can see here different carboxylic group like formate, acetate, and other higher amino acids. You can see that arginine forms the same pattern here, and the carboxylic groups here, or carboxylic molecules in the middle. So this pattern remains same irrespective of the change in the structure of the carboxylic acids involved. Professor Vijayan used to be punctual for anything for which he has agreed. It could be a meeting, a function, or submitting an evaluation or review report. Professor Vijayan often used to quote from Bhagavad Gita, Karmanyeva karyaste ma phaleshu kadajana. As you know, it says our duty is to accomplish a task most sincerely and not to bother too much about its results. In everything, he used to truly follow this. He had a total commitment to nation building. India will come first for him in every action. After joining MBU as faculty, he never visited USA even once till 1999. Even other foreign visits were also very brief, except for 1 year spent in Oxford, coinciding with the retirement of Dorothy Godkin. He was totally devoted to the development of crystallographic facility at IASC and establishment of macromolecular crystallography in India. If macromolecular crystallography is carried out in many research institutions of India, we owe it to Professor Vijayan. The only unhappiness he had was for not able to establish a world-class synchrotron facility in India. To some extent, he overcame this by arranging Indian access to European synchrotron radiation facility at Grenoble, France, and Trieste at Italy, and other places, and Japan also, through funding from DBT and DST. So this is a occasion arranged by Professor B. Gopal of MBU on the 75th anniversary of Professor Vijayan, birthday of Professor Vijayan. So we all assembled that time and seen here the cake, which has got all protein structures assembled here surrounding that, and Dr. Kalyani is helping Professor Vijayan to cut the cake. Dr. Kalyani was a great support for Vijayan all through, and we also couldn't separately think about them. Always we used to meet them together, or we are thankful for their hospitality at home also. Actually, I had some slides for explaining the crystallography, but I think the time doesn't permit, so maybe some other time I will talk to the audience for that. So with this, I conclude and showing some protein crystals. Thank you.
Thank you, sir. Thank you for giving a very close view of Professor Vijayan's career, life, personality, and research. And we understand how close you were with the family. Let me request you to continue in the platform. And now let me invite Professor Murthy, another much awaited talk. He was as Professor Suresh mentioned, he was professor at Institute of Science. He was a colleague for a long time with Vijayan, and currently he is at IBAB, Science and Technology Department of Government of Karnataka, as a distinguished chair and professor. And he has many awards, and including Shanti Bhatnagar Award. Without much delay, let me invite Professor Murthy, a very eloquent speaker and scientist in the field of crystallography. Professor Murthy, please.
Thank you. Do you see my slide?
Yes, I can. We can. Yes.
Okay, then it's all set. We are all set.
Sir, can you go to the full slide mode, full screen mode?
Yes.
Okay.
Is it okay now?
Yes, sir. Please.
All right. Thank you very much. You know, it's real privilege for me to speak on this occasion. I'm really delighted and grateful to the organizers. You know, what I want to convey, Vijayan's work is so massive that I won't be able to present all of it. But I would like to give a bird's eye view of his work, which I think non-biologists also should be able to follow. I want to tell Gurukkal that it won't be very difficult to follow what I am going to say, because I am going to avoid all technical jargon, because it's a mixed audience. So let's have it this way. You will have to bear with me, a slight overlap with Suresh. I didn't know what kind of presentation he is going to make. So there's a small overlap, but I will skip over that. Anyway, I had a 40-year association with Vijayan, and we almost meet several times every week over these 40 years. So I came to know of him extremely well, and I am so grateful for my work, my association with him. Well, this is what I'm going to skip. You see, this is what Suresh mentioned, that amino acids, which are really of which the proteins are made up of, they have a tendency such that when they are in a solid state, carboxyl and amino groups are proximal, so that they can actually bond. And this picture he also has shown, this also is part of what Suresh showed. All these pictures probably Suresh has already shown. Well, the conclusion from these studies, you see, Vijayan wanted to work on proteins. Even when, let me actually just go back in history and add to what Suresh said. This, macromolecular crystallography really started in the beginning of the sixth decade, that is 1960-62, for the years when structure of myoglobin and hemoglobin were worked out. Then in the middle of the decade, structure of insulin came out, in which Vijayan was involved. So when he came back to India, he was very keen on starting macromolecular crystallography, but there was no facility. So he had to work on something which has something to do with macromolecular crystallography. As Suresh was saying, examining amino acids and the interaction between them could be a model for what kind of interactions will contribute to protein stability and function. So he started on that. Let me actually share with you some other thing. When he was in Oxford, that was a time when crystallography was just coming out of age. And Britain was the center of crystallography. So any crystallographer trained in England got a plum academic position in the United States, because every university wanted to start macromolecular crystallography. But Vijayan had no second thoughts. His Karya Kshetram was India. So he would come back to India and start his work in India. So if he wanted to, he could have got job in any university in the United States. But that was not his priority. And also, you see, after spending some time in Oxford, when he came back, did he have an Oxford accent? People generally joke that no, the Oxford people had a Malayali accent after he stayed in Oxford for some time. He was that kind of a person, very original, very gifted. Anyway, what his structural studies on peptides and amino acids suggested is that these interactions, which are natural among these moieties, might have been crucial for really early evolution. That is, evolution of molecules before actually cells evolved. Well, of course, Vijayan himself was very aware of the uncertainties lurking behind chemical evolution. Actually, we don't understand how really life originated on Earth. Once the first cell was formed, we understand in a Darwinian sense how it can evolve into full-fledged human being. But how did the chemicals, so complex chemicals that make up a living cell, arise in an abiotic situation is something about which very little understanding is there. And of course, recently, for those of you who know the difference between proteins and nucleic acids, initially people thought, well, life might have started with proteins. Now the thought process is that it started with nucleic acid. Venki Ramakrishnan is one of the big proponents of this RNA world hypothesis, that life originated through RNA molecules. But anyway, this is extremely controversial, and it is part of what Suresh discussed. So let me skip over this. But,
Sir, can you switch on your video so that we can see you?
Oh, yeah, sure, I will.
Okay, thank you,
I
sir.
don't know how to do that. Oh, yeah, I can, yeah. It's on now. Okay. Fine.
Yeah, thank you, sir.
You see, actually, as I said, Vijayan was interested in macromolecular crystallography when he came back to India. But there was no facility. How can he contribute? One way was to work on peptides, which are part of protein, really, peptides and amino acids, which was part of proteins. But then can we do something else? He, along with his first student, which was shown by, he showed the names of P. Singh and T. N. Butt. He asked T. N. Butt to analyze side chain conformations of the few proteins for which the structure had been determined at that time. See, proteins have a main chain and they have branches, which we call side chains. And the side chains take some structures. What are the structures that the side chains take? That is the question. This can be statistically analyzed by analyzing protein structures. And then we can say, oh yes, this is the preferred conformation of such and such an amino acid. It will give us some idea about protein structure. All that I am saying, for those of you who are not biologists, in proteins, we have the main chain, and there is side chain. What are the shapes of these side chains? That is the investigation that Vijayan carried out along with his students, T. N. Butt. Today, this field is called bioinformatics. At that time, there was no term like bioinformatics. Also, it was a continuation of the rich tradition of Ramachandran School. So they actually published their research in 1979, when still MBU did not have any facility for macromolecular crystallography. This is the analysis of amino acid side chains in proteins, conformation of amino acid side chains in proteins. In some sense, this was a contribution to macromolecular crystallography, although there was no facility at that time. Then we got macromolecular facilities for doing protein structural work in the late 80s. Then Vijayan started on the structural determination of a lectin. Lectin are usually plant proteins with multiple functions. They do a lot of functions. They are biologically very important. But let's not worry about their biological importance. We will worry about the kind of structures they have. So Vijayan took up, unfortunately, the peanut lectin. Peanut is, we use peanut in every one of our food stuff. It has a lectin, which was crystallized in Surolia's lab, and Vijayan started crystallographic work on this. This is a very unfortunate choice. It really posed a lot of challenge in working, mainly because if you look at proteins in general, see, many proteins are made up of, every protein is made up of a polypeptide chain. But many proteins are made up of more than one chain, one polypeptide chain. For example, there is a protein here which has two identical chains, one shown in green and another shown in blue. This is a viral protein which has 4 chains. A single molecule has 4 chains. Here is a molecule which has 5 chains. Like that, you have different number of chains. Whenever a protein has more than one chain, generally these chains are placed very symmetrically. There is a two-fold axis of symmetry, which means if you rotate one of the subunits by 180 degrees, it exactly goes and falls on the other subunit. You see, you have a 4-fold axis of symmetry here. You have a 5-fold axis of symmetry. Like that, every protein molecule is symmetrical. And this assumption that a protein is going to be symmetrical was very detrimental for peanut lectin, because when they actually solved the peanut lectin structure, they found that it is made up of 4 chains, but it was not symmetrical. So actually this posed a lot of problems during structure determination, but eventually peanut lectin structure was solved. Once this was solved, the technology for solving structures was well established at Bangalore. Vijayan could take up a huge number of lectins and illustrate their structures. In principle, what he demonstrated is that, see, actually, this is a winged bean lectin. A winged bean is a dicot, and the lectin from this has a structure which we can call jelly roll motif. You see, this kind of structure where you have a lot of straight segments of polypeptide and another straight segment of polypeptide juxtaposed to each other in space. That has been called jelly roll motif. Such a motif is also found in viral proteins, many viral proteins like poliovirus protein. Why should these plant lectins have viral protein-like structures? We don't understand. But anyway, this was interesting. And then when Vijayan determined more lectin structures, he found different topologies in these structures. Just enjoy these pictures. Even if they don't make full sense right now, it doesn't matter. See, this pattern that you see here it's called a Greek key for whatever reason. See, the polypeptide chain is wound like this. You see, winding goes in a certain path, and that path is called Greek key. 3 Greek keys, tandem Greek keys, make up one lectin molecule. That way he could delineate the details of lectins. This is another, this is so-called beta prism fold lectin. This is from banana, the fruit that is most famous in Kerala, banana chips. There is a lectin in banana, and that lectin has a structure like this. Well, there is another, I don't see my own slide. Anyway, there is another lectin, see, in which there are 2 subunits related by a 2-fold axis of symmetry. But part of this blue subunits comes and gets associated with this brown subunit. And part of the brown subunit comes and gets associated with the blue subunit. This kind of phenomena is observed in many other proteins. and we call them domain swapping. It means these two subunits, these two polypeptide chains swap one of their domains between themselves. And there has been a lot of speculation that this might have been the reason why proteins have become symmetric. Proteins are symmetric. Whenever a protein has several polypeptide chains, they are arranged in a symmetric fashion. Why should there be symmetry is a big question for which we don't have the full answer, but partially people attribute sometimes this to, this kind of polymerization taking place through domain swapping. And Vijayan also has speculated what it means to have a lectin, domain swap lectin, or evolution. And there is another lectin which is so beautiful. This is called beta prism lectin. Just see the polypeptide chain, how it winds in space and makes a circular structure. Like that, I have shown here several lectins. All these are solved by Vijayan. Now, see, every protein structure determined is deposited in a bank, which we call protein data bank. So if you now enter protein data bank, it is maintained in, let's say, Europe, United States, Japan, and there is a mirror site at Bangalore also. Anybody can search and get data free of cost from any of the protein data banks. If you now look for lectins in protein data bank, it's very pleasing to note that the maximum number of lectin entries are from Vijayan. So if Vijayan's contribution to lectin structure is 50%, the contribution of the rest of the world is another 50%. So single man's contribution is equal to the contribution of all the other labs as far as lectin structures are concerned. Again, let me iterate what I said. Lectins are very important, biologically very important proteins. For example, if you want to determine blood group, A, B, O, you use a lectin. So lectins also can be used to deliver drugs to specific parts of an organism. See, the drugs that we take generally flood all our body, but the drug has side effects. If you can deliver a drug which has to act on liver, only to liver, this is targeted drug delivery. And such things may become possible if we utilize lectin structures. So these are very pharmaceutically very important structures. Although, of course, it takes the gap between fundamental discovery and application is very large. So I am not saying that these lectins are immediately useful, but someday somebody will use these lectins for very useful applications in the medical field or in many other fields. So that way these lectin structures are very important. And half the structures of lectins in the world are actually by Vijayan. In the late 80s, there was an accident in the lab. Somebody had put a crystal. See, this was an understanding that came from 1930s. When the first protein crystal was made, first protein was crystallized, they tried to shoot it with X-rays and collect X-ray diffraction data. Very soon, the crystal will stop diffracting. Then they realized that they are mounting the crystal and it is evaporating. All the solution around the crystal was evaporating. The crystal was getting dry. So they understood the importance of keeping protein crystals wet. You should never allow them to dry, they will go bad. So then onwards, technique was developed where a crystal was placed in a glass capillary with excess solution so that it will never go dry. And all the data were collected like that on wet protein crystals. But once by accident, I think it was Dinakar, he collected the data and crystal had partially dried. And lo and behold, the data was much better than the wet crystal data. So that gave an idea that, of course, if you dry a crystal completely, it will lose its crystallinity. But if you can control the degree of hydration, how much water content is there in a crystal may actually have transformation. This gave a very big, it started off a very big project on so-called water-mediated transformations of protein structures. So what does water do to protein structures? We are watery animals. In the sense, most of our body is made up of water. So in the presence of water, proteins behave one way. In the absence of water, they behave in a different way. What are these differences? Why do these differences come? These are important questions. So actually, Vijayan produced a series of PhD theses on this topic. What happens? So he took many proteins as prototype examples, like ribonuclease, hen-egg white lysozyme, lactoglobulin, and then partially dry these crystals. If you fully dry, of course, they will lose their ability to diffract. But you can control them. 95% humidity, 90% humidity, 85% humidity. What happens at each of these humidity values, the protein structure? That was a type of investigation. Now, because they showed that partial dehydration sometimes improves the crystal. It has become a standard protocol. If your crystal don't diffract, try partial dehydration. Maybe you are lucky and your crystals will diffract. This has been adopted in every crystallography lab in the world. Okay, now I want to tell you a number of projects on which Vijayan worked. I just want to, these are so detailed. Each one of the project has been done so thoroughly that we cannot cover everything about any of these projects. It's too detailed. I want to give a bird's eye view of how detailed analysis has been made on each one of the proteins that follow. See, a protein may crystallize in different forms. Same, you see, diamond and graphite are crystals of carbon. They are different crystal forms, but the material is the same, carbon. Like that, protein crystals also can come in different forms. We call different space groups, which means the way molecules are organized in space are different from crystal to crystal. So whenever proteins form more than one type of crystal, Vijayan studied every form absolutely thoroughly. And then proteins generally interact with smaller metabolites. Life goes on because proteins interact with smaller molecules and transform them. Maybe they will synthesize, they will degrade, they will isomerize. They do many actions in the cell. So you need to find structures of the protein along with ligand bound to the protein. So all these studies have been done in every protein they have selected for their studies. After doing a huge number of lectins, Vijayan shifted his interest to tuberculosis. Tuberculosis is a terrible disease of the third world country, and not many drug companies are very interested in developing a drug for tuberculosis because the rich countries probably don't buy this drug. They don't need it. But anyway, there has to be a concerted effort to tackle tuberculosis. This was firm belief of Vijayan. So he collaborated with a large number of biologists and microbiologists and, obtained clones of different tuberculosis. Tuberculosis is the organism that causes tuberculosis. There is another variant of the same bacterium, M. magmatis. It is not as pathogenic, but it has almost the same properties as M. tuberculosis. So both M. tuberculosis and M. magmatis proteins were studied by Vijayan. See, typically a bacterium codes for some 5,000 proteins. Our genome, human genome, probably codes for 20 to 30,000 proteins. Doesn't mean that we have only 30,000 proteins in our body, but what is coded by our genome is 30,000. More proteins can be created out of these 30,000 basic proteins. Anyway, let's say that we have 30,000 protein genes. In comparison, Mycobacterium tuberculosis has about 5,000 genes. Many of these genes, of course, all these genes are now at least known on paper, which are the genes coded by M. tuberculosis. But their structures are not known. It has to be determined by X-ray crystallography. What structure do you determine? So that is the idea behind determining structures from pathogenic organisms. The functions of many of these proteins are absolutely essential for the organism to grow and infect the host organism. So if you can knock off the function of any of these essential proteins, it will be a drug against tuberculosis. This is what we know nowadays call structure-based drug development. Earlier, most of the drugs, like Ayurvedic drugs, for which Kerala is famous, most of those drugs were serendipitously discovered. Somebody ate a plant and they had terrible diarrhea. So they know that the plant has a substance that causes diarrhea. Like that, accidental discoveries gave us many drugs. All the earlier drugs have been discovered by accident. But today, because we know how proteins function, we can actually use our knowledge to develop drugs. This can be molecular structure-based drug design. It was the idea Vijayan had. If you know the structures of key proteins of tuberculosis, M. tuberculosis, Mycobacterium, then maybe it will lead to development of drugs against tuberculosis. That is the idea. And then you have to select the targets carefully. Which are the proteins that are very important? So actually, there is a protein called Rakie. In every case, Vijayan and his students have solved the structure of the protein as far as possible, both from Mycobacterium tuberculosis and Mycobacterium smegmatis. See, in terms of evolutionary terms, we can think of M. tuberculosis and M. smegmatis as very close cousins of each other. Their genomes are very similar, the proteins are very similar, the structures are very similar, the mechanism of action, the mechanism of infection are very similar. So a comparative study of both tuberculosis and smegmatis will give us a richer repertoire of information than studying any one organism. So they have studied both the organisms. And then they have studied mutants of these proteins. That means if you change key amino acids, what kind of changes take place in the protein structure? And then how is the function of the protein affected by these mutations? And what ligands bind to these proteins? You can ask many questions on protein structure. Every aspect of it has been worked out for a large number of proteins. So in some sense, I have just summarized what was the purpose of studying these tuberculosis, M. tuberculosis proteins coded by Mycobacterium, and how the targets are chosen. The targets are chosen because those proteins play a key role in the life cycle of Mycobacterium. So with that, I will just go through the list of proteins which has been studied and what kind of insight it has provided on mycobacterial infection. So one of the proteins, only for this protein, I show a little bit. This protein is called Rakie. This is necessary for DNA repair. Let us say when DNA is damaged, if it has to be repaired, this Rakie plays a very important function in managing DNA, which is the genetic material of cells. So this Rakie protein happens to be a very key protein for Mycobacterium. Now, if you see what is shown in blue here, see, proteins are so complex. See, let me actually, for those of you who are not all scientists, those who come from literature or other fields, water is a very simple molecule, which is made up of 2 hydrogen atoms and 1 oxygen atom, only 3 atoms. Whereas proteins have thousands of atoms, sometimes tens of thousands of atoms. If you show all these atoms, it will be very confusing. It will be a forest of atoms. It doesn't make sense. So the way we illustrate protein is that because it's a linear polypeptide chain, we show how it is folded in 3-dimensional space. Now, I have shown in blue here the Rakie protein from Mycobacterium tuberculosis. In gray here, Mycobacterium smegmatis Rakie. You can see that structures are very similar, yet there are differences. So you can ask what is the significance of these differences. So a whole lot of questions you can answer. See, they have also examined surface features. If you look at the surface of the protein, how rigid the surface is, or how poly the surface is, are there cavities on the protein surface, and whether there are charges, electrical charges on the protein surface, or it is electrically neutral. All these examinations have been done for every protein, and I am not going to show in other proteins. See, if a protein binds a ligand, just see how they are made for each other. You can call it lock and key. The protein here, what is shown in white is the protein, but it is shown only in surface representation. And there is a ligand here, which is shown in atomic representation. You can see the structures snugly fit into each other. This kind of studies has been done on every molecule, whatever molecule I am going to show. So actually, there is a protein which actually unwinds DNA. DNA, our genetic material, is a double-stranded molecule. There are two strands. And during many processes of cellular function, the strands have to be separated. And to separate them, there are a set of proteins. They are called single-strand DNA-binding proteins. And one of those proteins from Mycobacterium was determined in Vijayan's group. And I have shown different views of the molecule. You see, actually, this is a domain-swapped molecule. What I was referring to earlier, that part of the green subunit wraps around this subunit, and part of this subunit wraps around that subunit. And Mycobacterium's magmatis protein, the 4 polypeptide chains are organized in one way, but in E. coli, the same protein is organized in another way. So there is a change in the quaternary organization of the protein. What is the implication of this? You see, E. coli also is a kind of pathogenic bacterium, but not as deadly as Mycobacterium. But there is a difference in the protein from the two organisms. Of course, E. coli structure was determined in some other lab, whereas Mycobacterial structure was determined in Vijayan's lab. Let me also add that each of the pictures I am showing is the subject matter of at least 2 PhD theses in Vijayan's lab, and perhaps 1 or 2 theses in his collaborator's lab. So what we are actually skipping through each slide is 3 or 4 PhD theses. So obviously, what I am telling you is exceptionally brief description of what has been achieved. Well, you see, uracil is a base or a letter in the DNA genome of our genes. Uracil is one of the nucleotides that occurs in the nucleic acids, and that uracil can actually undergo modification. And there is a protein associated with this modification, and that structure is what is shown here. And then you see, actually, ribosomes are the machinery on which proteins are synthesized. I cannot give you more details. If you are a biologist, it is too obvious for you. If you have never studied biology, no matter how much I explain, I am not going to make it very clear. But ribosomes are the machinery on which there are factories on which proteins themselves are synthesized. That ribosome function has to be controlled by few other proteins. This is one of those proteins is actually the structure of which was determined in Vijayan's lab. This structure can exist in one form like this, where this domain is organized almost perpendicular to the other protein domain, whereas it can undergo a very large conformational change. You see, after all, if we want to work, we have to move our limbs, right? Otherwise, we cannot move, we cannot walk. If we are a stone statue, we cannot do anything. Similarly, if proteins have to carry out function, there has to be flexibility in the protein. What are the flexibility in a given protein? In what way it is flexible? This also becomes a very important question. And here you can see that they have shown what kind of flexibility protein can exhibit. Well, actually, this is another protein, a protein that breaks covalent bonds. So actually, as I said, I will not give you more descriptions of these. Just please note that each one of these proteins has been absolutely thoroughly investigated, and each protein is very key to the life cycle of mycobacterium. So potentially any of these molecules can be a candidate for development of antimicrobial drugs. So there are more proteins. As I said, I don't want to spend time on it because you see each one of these is a big story by itself. And this is yet another protein. See, in our own lab, we had worked out the structure of some other proteins, and this protein worked out in Vijayan's lab accidentally has some similarities to some other protein we had solved. Well, of great interest is some of the very complex protein structures that Vijayan determined. You know, this is in collaboration with Dipankar Chatterjee of MBU. You see, bacteria, mycobacteria particularly, you know, almost all of us are infected with mycobacteria. Whether you know it or not, we harbor mycobacteria in our body. But we don't have TB, you see. But when our immune system is weak, the bacteria that is hiding in us may start actually causing tuberculosis. So most Indians are actually infected with TB. If you really make a test for presence of bacterium, you probably find in everybody. But how does it survive for a long time without causing infection? This is what we call survival under stress conditions. You know, even if you put mycobacterium under great stress, it somehow survives. And when the opportunity arises, it starts multiplying. And this process has to be understood. In this, actually, there's a protein which can actually 12 of the polypeptide chains can assemble into what we call a dodecamer. Dodeca means 12. 12 chain polypeptide, or it can actually go into a 24-mer. These are some of the very big proteins, and they also, Dipankar also has demonstrated that these proteins can actually soak up iron atoms. So that, you know, if you want to take away iron atoms from a cell, you can have more of these proteins. So this can be related to how bacteria survive stress. These structures are some of the largest proteins determined by Vijayan. Well, I just now have the last of his slides, last of the protein, I mean, the list of the proteins on which Vijayan worked. All these proteins Vijayan has worked. Somebody else has worked on these mycobacterial proteins. It is really very, very large. So I just want, as my dear colleague and friend, I just want to give you a brief idea of Vijayan as a person. Actually, Suresh has done justice to this. He has said how warm Vijayan was. You know, Vijayan, I always like to think, comes from the God's own country where lots of coconuts are grown. If you break coconut, it has really mineral water, refreshing mineral water inside, and coconut, which goes into making every sweet. But outside, it is very rough and tough. Like that, Vijayan was really rough and tough. He could take anybody to task, including financial controller of Indian Institute of Science. You know, he wouldn't hesitate to call him and say, what you are doing is nonsense. This cannot be done. So that way he was very rough, but inside he was very soft. You know, I like to compare him with coconut. There are other fruits which look very smooth outside, but there is nothing, no thing inside. This, I think Suresh has done a very good job. I am only supplementing it with a figure. You know, he said how Vijayan stayed out of a dinner party because drivers were not allowed. So I think this is a very natural setting to see Vijayan with really socially unfortunate individuals. You know, he had such large heart for them. It came out in every one of his actions, one of which was mentioned by Suresh. And I also want to make a joke on Vijayan to say that he was a bodybuilder, a big bodybuilder, mainly because, let's see, this is India, our India. And you see, there are X-ray labs now in India. First was Bangalore and almost simultaneously was Bombay. But now you see the number of labs that are there in India. So many X-ray laboratories. And most of them is because Vijayan bulldozed DST to support X-ray labs in all these places. This is something of a very generous thing, not to get facilities to MDU, IISc, but to see that every person who is capable of doing research gets his facility. See, many of these people are not as big and recognized as Vijayan. So if they had written grants on their own, maybe DST would have given, maybe they would not have given. Vijayan was instrumental in most of these establishments. He gave very good advice to people as to how to go about applying to DST grant, how to get the grant, and how to utilize the grant to establish the lab, and how to work very well. This also actually has been covered by Suresh. How to cajole people to work. Vijayan was very good. He would be having 8:30 in the morning and expect his people to be there. So you have to be truthful to others, and then you have to be truthful to yourself. Doing what is right, regardless of who is around. That's very important, and it needs a lot of courage. Although it's easily said, it needs a lot of courage. And work honestly. All these qualities were with him, so he could accomplish so much. And one of the things I wonder very much is the number of collaborations he established. He could do all those mycobacterial proteins because he collaborated with so many biochemists and microbiologists. All these different people had their own personalities and, idiosyncrasies. To work with all of them needed some kind of balance and a very mature personality. That's why I said some of the collaborators were toads and occasional scorpions, but Vijayan handled all of them extremely well. So he was kind of international person, but never without self-respect. So whatever he did had to be done in India, and it has to be an Indian achievement. He was very clear about it, but he was very international. In some sense, he didn't have national or state barriers. Although you people want to claim as a Keralite, he's an Indian, he's a world citizen. So that way he had no differences. But when we achieve, it should be ours, not because some white man has contributed to it. Last word I want to have is, I have shown what are the kinds of things you could eat in Vijayan's home, with Kalyani Vijayan supervising everything. And the way Vijayan talked to Kalyani was so full of love and respect. Conversation appeared to be background music to their home parties. I have gone to their home in the presence of greats and students, and every occasion I have enjoyed very much. The only thing that makes me very sad is that he lost his health. His mind was extremely sharp, but physically he became almost invalid towards the end of his life. It broke my heart so much that I didn't have heart to go and see him. So that kind of speaks for my attachment to Vijayan. With this, I conclude my presentation, and thank you very much for allowing me to join your group and make my little contribution in honor of my colleague, Professor Vijayan. Thank you very much.
Thank you, sir. Thank you very much for your excellent talk, which could account his research in a layman's language. A person from different field could also feel what was his research and how important it was. And then lastly, his personal aspects. And some of these traits are very important to know. And I think, we can really follow some of these things. And now, at last, and let me invite, so we could already hear one of his first students and his very close friend. And then now he's one of his latest student, Professor Saikrishnan Kayarat. He's a professor at ISER Pune. So he did PhD with Professor Vijayan, and then postdoctoral research at UK, and then came back. With all respect, let me invite Professor Saikrishnan for his brief talk. Please.
Thank you, Dr. Narayanan. Thank you, Satishan, for inviting me to this commemoration that is celebrating Professor Vijayan's scientific achievements. it's a great honor and privilege to be among such eminent speakers. And it's a wonderful occasion for me, and something that brings back memories of my interaction with Professor Vijayan. I will be very brief. I'll present my slides. I'll be very brief about what I'm talking about. It's what I'm going to present is a student's tribute to Professor Vijayan. I was a PhD student, not a very recent student. I was a PhD student with Professor in 2000 to 2005. I joined IISc as an integrated PhD student. I worked in Professor's lab for 6 months as a rotation student, and then continued working there as a PhD student. The lab environment was primarily what made the decision so easy for me to just continue working there. It was a home away from home for all of us in the lab. And an environment of camaraderie was present always there in the lab. And I joined as a PhD student. Professor became the associate director of IISc Bangalore. At this moment, I and my lab colleagues felt, okay, possibly Professor is going to be busy with his administrative duties. Maybe the lab is going to have some free time. Maybe we can have some joy time. But Professor was there bang at 8:30 the next day morning, asking us what our work has been, discussing the scientific progress that we have made. And to be frank, these discussions have been the training grounds where I learned, I bit my crystallographic teeth. And this is what has been one of the most important pillars for the scientific research that I've been carrying out in my independent career at IISc Pune. It was the same year when Professor Vijayan initiated the Indian Tuberculosis Structural Genomics program, about which Professor Murthy just mentioned. And and this was an important component of the International TB Structural Genomics program. So Professor Vijayan had the vision of a structural genomics program for tuberculosis, and simultaneously an international program also came up, and we got, it was a holistic absorption of the Indian effort and the international effort that resulted in understanding the structures of a large number of proteins, that 3D structures, which, as Professor Murthy mentioned, why they are important, their importance to understand their function, to develop drugs. These are the requirements that could be facilitated with the structure determination of these proteins from the pathogen Mycobacterium tuberculosis, which is the causative organism of the disease tuberculosis. I joined this effort in my PhD work. I determined the structure of single-stranded DNA binding protein and ribosome recycling factor. Some of these two proteins were shown by Professor Murthy. It was an honor to see Professor Murthy show the structures that I determined for my PhD, and I'm very thankful to him for that. And as Professor Murthy said, it's not just me. A large number of students in the laboratory over the years determined structures of proteins from Mycobacterium tuberculosis, primarily in those that are involved in replication, recombination, repair, DNA replication, recombination, and repair, and translation, and certain biosynthesis processes. But what was important, more than just starting this initiative, was Professor's thrust on making this a pan-Indian program where research scientists working on tuberculosis collaborated and overcame any kind of barriers that they would have, either physical separation in distance or getting reagents and so on. All these barriers were overcome, and India contributed extensively to this program, and it became one of the most successful programs where Indian contribution to the international effort has been substantial. And what I'm showing here is not only structures of mycobacterial proteins determined in the laboratory of Professor Vijayan, but some of them that were determined in other laboratories also. And this is just a partial list of or partial picture of the proteins that whose structures have been determined. There are many more, and this contribution is an important contribution from the Indian scientific community to the world of tuberculosis and our efforts to overcome this disease, the challenges that it poses. While these efforts were going on in the laboratory and the country, Professor was also relentlessly pursuing the establishment of state-of-the-art crystallography facilities. Again, Professor Murthy showed how the structural biology laboratories and crystallography laboratories bloomed in different corners of the country, starting with Bangalore and Bombay at BARC, and then going on to cover the entire, almost all the states and cities or important cities of the country wherever there are scientific institutes. And most of these groups came from, were established by students who worked either directly with Professor Vijayan or were part of the molecular biophysics unit, like students of Professor Murthy, and others who were part of what Professor Vijayan would call a bigger gharana of Indian structural biology. And I should say here that the MBU community, in particular the crystallography community, was a close-knit community. While I learned a lot of my crystallography directly from Professor Vijayan, I was greatly influenced and learned many of the state-of-the-art techniques from Professor Murthy, who was excellent with his programming skill and knew a lot about the new programs that were coming out. And we would always go to him to learn about this, to discuss the challenges that we are facing. And that made our work seem so effortless. At the same time, getting us learn new things that would not have been possible, even, I guess, abroad, because I've seen laboratories abroad where there are crystallographers whose students may be learning crystallography, but focus towards just solving the structure that they require to solve it, but not learning the intricacies, the nuances of crystallography, which I think is something that is unique to molecular biophysics unit at IISc Bangalore, and something I hope grows and is growing in other parts of India as well. Finally, I'd like to conclude my tribute by quoting Professor Balram, who in his obituary in the journal Current Science wrote, "In his remarkably productive career that spanned over 5 decades, Vijayan was a researcher, teacher, and administrator who greatly influenced his surroundings." And this is from a person who worked closely with Professor Vijayan and who has known him for a very long time. And with that, I'd like to end my tribute. Thank you very much.
Thank you very much, Professor Saikrishnan, for your recalling those loving memories and again emphasizing how important he was as a person and also his research. Now, let me invite participants who are very close to Professor Vijayan. I know some very special guests are there. Can I call? I think I understand that his daughter Devayani is there. If possible, madam, can you share your valuable memories? I know it's quite hard, but this will be a very valuable moment for us. Ms. Devayani Vijayan, can you hear? Can you share your?
Yeah, no, I wasn't able to unmute myself for a second.
Okay. Okay.
Wait, let me. Thank you all so very much for all these wonderful memories. And like we had said in another occasion, this is celebrating his life in a sense more than anything else. And the outpouring and love and affection that we have seen over the last few weeks have been tremendous. And I think a large part of it is the joy love that he got back reflects in how he said it to everybody else as well. So it is a combined thing. It's not a one-way street. It is what you give is what you get. So thank you all very, very much.
Thank you, madam, for your valuable presence. And your mother is around with you, or she can
She
share
is
some?
actually, we are both in separate locations right now. But
Okay.
I don't know if she wants to talk. Amma, do you want to speak?
No, not really. You have spoken.
All right. Okay. I have
Not
represented
really.
both of us.
Okay. Okay. Thank you. Thank you very much.
Thank you.
Thank you. Thank you for your presence. And now let me invite from the participants. Please unmute yourself and share your valuable memories of with Professor Vijayan. That would be very great for this occasion. I don't want to name anyone. Please unmute yourself who are very close to him. Or if not, I request the participants if you want to. Yeah.
This is Rajashekar.
Yeah.
I'm not a direct student of Prof. Vijayan. I am from IP's department, but I knew Prof. Vijayan very closely since the time I joined in 1977 till I left the institute in 81. See, the thing is like that. It is very hard on my part to give a tribute to Prof. Vijayan, thinking that he's no more with us. Okay, it is a great loss to chemistry. Okay, in general, I should say, and particularly the students of molecular biophysics and macromolecular crystallography will be really orphaned. Okay, but again, your students should take it forward where he has left, so that the legacy continues. And if the people we love are stolen from us, the way to have them live on is to never stop loving them. This is what James O. Barr said. Okay, I really, really love everybody to follow this one, and let us keep the flag flying. Okay, yes, hoisted the flag for us. Okay, let it keep flying in very high, and let us contribute whatever we can make it. Okay, I love this family, and my Prof. Vijayan, whenever I go to MBU department, he has always has a soft corner for me to invite me to his room. Okay, so I'm grateful to him that way. But one day I will meet him in the heaven. Okay, he'll be waiting for me there. Thank you very much. Let the soul attain Atma Shanti. Let us pray for him. Om Namo Narayanaya. Thank you.
Thank you, sir. Your loving sharing us share with your loving memories. So, anyone from the participant, please unmute yourself. And also, we request you to switch on your video too. Prasad Narayan Kurur, you want to share your memories?
Hello, can I say something?
Please.
Yeah, I am Desh Deepak Singh, and I was his postdoctoral student, and Saikrishnan and me, we were all together at that point of time. And one point I just picked up from Professor Murthy is that is a analogy he gave to coconut. Okay, he knew the parameters, but with a very friendly atmosphere, he would let you play in that parameter. Right? This is something very, very. Now, when we are teachers, when we are teaching our students, this is an extremely important trait. You know the boundaries, but let the kids play in that boundary the way they wish to play. This is something wonderful about Professor Vijayan I want to share with all. And he would give you a family atmosphere. We could go and crack jokes with him when he go to the house, and ma'am is there and Devyani is there. We just thought we are a part of his family. You know, we all came from very far off places, and he would see to our welfare, being there in his lab, which was wonderful. The ma'am will, make us feel at home, away from home. But that analogy of the coconut stays with me. He showed us very nicely that this is the parameter friend. You have to work hard, you have to be productive, but play as much as you can within these parameters. So what that essentially helped us was to really evolve not only as a scientist, but I would say also as a human being. And we are carrying those traits. And the last thing I would like to just share is that I have said many occasions that there is a word in Hindi called somebody who can touch and he becomes gold. So he had that particular capacity with him. I think we owe a lot to him. He touched a lot of raw people and turned them into gold. And that is why you see a plethora of his students spread across the country, not only in India, but across the world. who are doing so wonderfully well. I mean, I don't feel that he's not there. I really don't feel that he's not there. I can always feel him in my work with me. So I'm not sad that he's not there. But what he taught us lives with me, and that's more important. So I always feel his presence and his values and thoughts. And of course, we look forward to now ma'am and Devyani to carry on that same connection. I look forward to meeting them on 20th. We are also coming to ISC. There's some program there, so we'll meet them. We wanted to meet them very, badly, really. So I hope we can all get together there. Thank you very much, Professor Suresh, Professor Suresh, and my dear friend, and all of you to give this opportunity. Thank you very much. Bye.
Thank you, sir. Thank you. Thank you for your valuable words and memories. Now, I request the other people who are very close to him to share their memories. Or if anyone wants to pose any questions to any of these speakers, we can also use this occasion. So, if not, again, let me come to the end of this.
Narayan, one second. Narayan, one second.
Yes, please.
No, actually, I just wanted to add what Devani is saying. When we celebrate in Vijayan, actually, it's not just we are honored. Science people could organize such an event, and eminent guests were there. We are really thanking them. So it's really a good moment for science people to meet all of you here and hear you everyone very closely about Mr. Vijayan. Yeah, it's really just wanted to that science people is really thank you for this.
Yeah, so this is the entire motto of the Science People platform, to bring scientists close to the community and start interacting with them so that we will know more about their contributions to the society and to the science and scientist as a person. So thank you all the speakers, giving their valuable memories with Professor Vijayan, and various aspects, as I mentioned in my initial introduction, as a scientist, as a person, as a teacher, as a passionate researcher, etc., etc. But we almost spent 2 hours, and once again, thank you for all the speakers for wonderful memories and sharing with us, and also a special thanks to Madam Devyani and Kalyani, Madam Kalyani, for being with us in this program. That's really very thankful to them. Now let me invite Mr. Abhishek Shivan for giving vote of thanks. Abhishek.
Yeah. Thank you all for joining this program, and we are very grateful for all the speakers, eminent speakers today joining this platform, and also special thanks to T.N. Narayanan for moderating the session, and once again very thanks on behalf of science people to all the friends and eminent speakers joining this session. Thank you very much.
Thank you.